Everyone, I just wanted to finish up chapter 5 for you. I'm coming to you from a patio, so you're going to hear birds in the background. Currently, it is completely blue skies, but I guess we're in for some showers today. So if I stop the recording quickly, you'll know why. All right, so we left off with metabolism of lipids and proteins. And overall, fats probably have the greatest amount of energy stored, while proteins have a little bit. The important thing is we can use both to make ATP. So fats are hydrolyzed glycerol and fatty acids, and it's the fatty acids that can enter directly into the Krebs or citric acid cycle. Now, the glycerol itself can enter glycolysis. Now, proteins, when they're broken down, they break down to amino acids. They can be deaminated, and they enter directly through the Krebs citric acid cycle, like the fatty acids. All right, we have energy storage, which is really important. When more energy is taken in than it's consumed, we inhibit ATP synthesis, or our bodies inhibit ATP synthesis. At this point, glucose is converted into glycogen as well as fat. And this is showing you exactly the process. This flowchart is showing you the exact process of what's going on. And so think about it, right? You can take glucose here and add a phosphate. Once you add the phosphate, it's converted from glucose 6 phosphate to glucose 1 phosphate, which is then stored as glycogen. And then the opposite can occur should we need energy. And you can see that other arrow here. And glycogen can be broken down to glucose 1 phosphate and then glucose 6 phosphate and all the way down to eventually become acetyl-CoA. All right, fats. Now, too much ATP. We don't need all the acetyl-CoA. We don't need the 3-phosphoglyceraldehyde. So the 3-phosphoglyceraldehyde becomes glycerol, and acetyl-CoA can convert to fatty acids. So you have the glycerol, the fatty acids, and there you have the storage as fat. All right, acetyl-CoA is pretty amazing in the fact that it can have 4 different pathways depending on what's needed by the body. So the one we're most familiar with is this pathway, because that's what we've been talking about when we talked about aerobic respiration. And this is going acetyl-CoA entering the citric acid or Krebs cycle, and then eventually CO2 is the product of aerobic metabolism. And then the next one is acetyl-CoA converting to fatty acids, and I showed you that on the previous slide. And fatty acids can be broken down then to phospholipids and triglycerides or triacylglycerol. Acetyl-CoA can also become ketone bodies. Now, ketone bodies can be really important in energy because the brain requires either glucose preferred or ketone bodies for energy. That's all they can use for energy. So if there's little glucose around, acetyl-CoA can be converted to those ketone bodies to keep the brain functioning. And finally, acetyl-CoA can be converted to cholesterol. And cholesterol, of course, can be utilized for steroids as well as for bile acids. And we'll talk more about bile acids when we get to digestion. All right, lipogenesis. Acetyl-CoAs, of course, can be linked together, and when they're linked together, they form fatty acids. So you remember that the fatty acids plus glycerol become what's called triglycerides. And this is where it occurs is mainly in adipose as well as the liver. This is how individuals sometimes get a fatty liver. Fat itself is, like I mentioned before, is a major form of energy storage in the body because it yields 9 kilocalories per gram. Now, carbs and proteins only yield 4. And we're actually going to delve deeper into this when we do food and exercise physiology. Okay, lipolysis or lipolysis is the breakdown of fat into what we know as fatty acids and glycerol, and it's via hydrolysis using the enzyme lipase. And of course, we're going to get more into the enzymes when we talk about digestion. Acetyl-CoA now from the free fatty acids. Now that serves as the major energy source. And we've already looked at that picture showing all the ways it can become energy sources. Right, beta oxidation is an interesting concept, and what it is it clips acetyl-CoAs off fatty acid chains as needed. So think about it this way. For every 2 carbons, okay, there's that 2, on a fatty acid chain, 1 acetyl-CoA can be formed. So think a 16-carbon fatty acid, divide that by 2, you get 8 acetyl-CoAs. Now, acetyl-CoA, each one results in 10 ATP. And we're just talking about the Krebs cycle. So 16-carbon fatty acid, 80 ATP. So make sure you can do this conversion. It's just division by 2 and then times 10. Right, so 8 times 10 gave us that 80 ATP from Krebs cycle alone. So that's a lot of energy. Okay, those ketone bodies that I mentioned prior, sometimes when the rate of lipolysis exceeds the rate of fatty acid utilization, in particular, dieting, starvation, or diabetes, and typically the dieting would be, the keto diet that's kind of a fad right now. But this is where the concentration of fatty acids in the blood increases. So the breakdown of fat into acetyl-CoA, right? And then the acetyl-CoA takes the pathway of ketone bodies. And again, it can be used for energy, like I said, mainly in the brain. Ketone bodies are water-soluble molecules that circulate in the blood. But here's the problem. Ketone bodies are acidic. So every time you have this increased conversion from acetyl-CoA to ketone bodies, it leads to a buildup, and it causes what's called ketosis, or even more, like I said, they're acidic, ketoacidosis. And that's seen in type 1, sometimes type 2, but mainly type 1 diabetics that are breaking down fat for energy because they can't use the glucose. And it can be pretty dangerous. This is an ICU leading complication of diabetes. And personally, having had type 1 diabetes since I was 9, and back in the olden days, I've had ketoacidosis about 4 to 5 times, and pretty traumatic. All right, amino acid metabolism. Now, nitrogen is the key here. We ingest nitrogen primarily as protein. And nitrogen, of course, is then used in amino acids in the body. If we ingest too much nitrogen or too much protein, the excess actually is converted into urea and is excreted in the urine. The kidneys also use urea as an osmotic to help with osmosis of water out of the urine. So we end up either with a positive nitrogen balance or a negative nitrogen balance. So remember, if you ingest more nitrogen than excreted, it's a positive balance. If it's less nitrogen ingested than excreted, it's negative. So in a healthy adult, the amount excreted equals the amount ingested. Now, excess amino acids from a positive nitrogen balance can also be converted into carbs as well as fat. Now, there are essential and non-essential amino acids that are utilized in the body. There's 20 total, so 20 amino acids total. 12 of those, no problem, can be produced by our body. However, 8 comes from the diet, and we call these essential amino acids. They include amino acids such as lysine, tryptophan, phenylalanine, threonine, valine, etc. You do not need to memorize all of the essential versus non-essential, but you should know these numerical values. Now let's talk about transamination. Transamination is when we take the intermediates, such as pruvic acid and citric acid intermediates, specifically keto acids, and convert them into amino acids. And it's just simply by adding the amine group in H2. So one of the things I want to draw your attention to is the enzyme that's utilized to convert into these intermediates into amino acids or different amino acids. So the first is AST, that's aspartate aminotransferase, and ALT, that's alanine aminotransferase. And they might be familiar to some of you because these are liver function tests. And so if there's ever a question of issues with the liver, they'll run liver function tests. And these are the two enzymes they look for to see if they're elevated. Just a side note, ALT is what is looked for an individual that drinks alcohol heavily. And ALT often rises during that time. And I always remember that because AL, right, alcohol, ALT. So what we have here is glutamic acid binding with oxaloacetic acid, and we end up with aspartic acid. Makes sense. Same thing, glutamic acid, but this time we're going to add pyruvic acid. Previously, it was oxaloacetic acid from Krebs. And we get another intermediate of Krebs, alpha-ketogluteric acid, and we get alanine, so 2 amino acids. Now, oxidative deamination. So if there's more amino acids than we need, the amine group from the glutamic acid is stripped and excreted as urea, when we talked about urea in the urine. So what happens is the amino acid becomes a keto acid, right, via transferring the amino group. And what we get during this transfer, there's the amino acid, the alpha-ketogluteric acid. Sorry, my drawing's not very good. We get that amino transfer to form glutamic acid. Off of glutamic acid, we end up with ammonia and water, which becomes urea, and there's the structure of urea. If you're curious, you don't need to learn the structure. Okay, this is pretty much our last slide, use of different energy sources. And it's important that you note a couple of these. The first is the brain, and I've already said this. The brain is glucose pretty much only. But once glucose reserves are gone, we'll accept ketone bodies. Now, the skeletal muscle, when it's resting, fatty acids are the main source. Okay, main source. Liver, fatty acids, but can also utilize ketone bodies. In fact, the liver can utilize lactic acid, glucose, fatty acids, and ketone bodies. So all of them. Same as the heart, which is pretty important to maintain cardiac function. Ideally, fatty acids, because of course they give the most energy, the ATP, but the heart can also utilize lactic acid, ketone bodies, and glucose. So this list is an important list of what is ideally used by each of these four organs and what can be used, and what can't be used. So take a look at this before you take your exam, and keep in mind some of the key points, mainly that the glucose and ketone bodies is the brain. Skeletal muscle is mainly fatty acids, but not lactic acid. Liver can use all, and heart can use all. And I think that's our last slide. There's a Kahoot. We didn't do it because I actually have your quiz to assign your participation credits. So everyone have a fabulous Sunday. I will see you tomorrow. I'm excited to start chapter 6. Bye-bye.